Today we'll be talking about the cell cycle. Ah! All right, let's have a go at doing the 3 quick questions. We'll have a chat about them in class together. In your study design, we're doing the very start of Unit 2, Area Study 1, which is all about the cell cycle, something you guys would have looked at in your holiday homework, hopefully. And the cell cycle is all about the derivation, so getting all of cells from pre-existing cells through the completion of what we call this cell cycle. Okay, we're going to talk about the cell cycle in terms of prokaryotic cells and in terms of eukaryotic cells. So prokaryotic cells is really simple. It's just a process that we call binary fission. Eukaryotic cells is a lot more complicated, and we have phases of interphase, we have phases of mitosis, and we have something called cytokinesis, which comes in between. Alright, so there's 3 different aspects that are important in terms of the cell theory, and here's the 3 right here. Just let it restart really quick. So the very first aspect is that all organisms on Earth are composed of 1 or more living cell. Number 2 is the cell is a basic unit of structure and organization of all of those organisms. And number 3, most importantly, is that every cell comes from a pre-existing cell. You can't just get cells, poof, out of nowhere. And it's actually one of the biggest mysteries of science and of biology is how did we get those first cells, those first bacterial cells? So let's first talk about prokaryotic cell division. So prokaryotic cell division is all about what we call binary fission. Okay, and this is the process of binary fission right here. All right, binary fission is pretty much results in the replication of 1 single cells into 2 cells, and then 4 cells, and then 8 cells, and then 16 cells. We're just doing 2 to the power of n number of divisions. And each of those daughter cells is going to be an exact clone of that mother cell. Well, nearly an exact clone of that mother cell. And here's an example of how crazy, this is sped up, but binary fission gets pretty nuts, and it's the reason why we have so many bacteria in the world. 3 very simple steps to binary fission. All right, here we have a singular bacterial or prokaryotic cell. The first thing that's going to happen is that DNA, which is circular, is going to simply replicate. So here we have 1 copy of the DNA, and that turns into 2 exact copies of that DNA. The cell is going to elongate, so it's going to get a bit longer and longer, and the middle is going to pinch off, and the cell membrane and the cell wall of the bacteria is actually going to pinch off and split. All right, so pretty much these 2 membranes is going to eventually split, forming 2 identical daughter cells. The only difference is this thing that we call plasmids, and in this picture it's not represented, but pretty much these little tiny circles of DNA are actually just split into the 2 cells. So for example, this cell on this side might get a few of the plasmids, this cell on this side might get some different plasmids. Okay, that's binary guy of cell division, which is eukaryotic cell division. The eukaryotic cells, they have to, because they're more complex, they have to use a more complex process, and we call this process mitosis. Right? And mitosis is the process where all cells, except for sex cells, which we call gametes, which we're going to look at later, replicate to form new cells. It's a process which involves a diploid cell, which we call a 2N cell, and that's in humans. So humans have 2N cells, dividing into 2 identical diploid cells. Okay, what we mean by 2N or diploid, it means that we have 2 sets of chromosomes, which makes sense because we get half of our chromosomes from mum and half of our chromosomes from dad. How often does this process of mitosis happen? If you snap your fingers, pretty much every time you're snapping your fingers, that's 2 million red blood cells dividing in your body. It's also 2.5 million dying, but regardless, you're getting 2 million red blood cells dividing through this process of mitosis. This is the cell cycle, and this is a very important diagram for you guys to have, okay? And you can actually run a cell through the cell cycle and to see how it divides and what happens. So let's say we start a cell right here at the very start of what we call the G1 or the gap 1 phase, and that cell will go through the G1 phase. During the G1 phase, what it does is it grows and it just does its normal protein building processes. If that cell is going to go divide, it's going to go through the S phase. And what the S phase is the synthesis phase. In the S phase, the chromosomes of the cell go from being linear single-stranded chromosomes to replicating and becoming double-stranded chromosomes, and it's just made up of 2 identical chromosome strands, and that's going to be very important later on. After that, the cell undergoes what we call the gap 2 or the G2 phase, where it just does some more growing and some more protein building, and then finally, when it's ready, it goes through what we call the M phase or mitosis. And in mitosis, what's going to happen? We're going to have an extra perfectly identical daughter cell. All right, so we're going to go from 1 mother cell to 2 identical daughter cells. And then that process is going to cycle through again. All right, and once it's gotten back, each of these cells will have produced 2 identical daughter cells. And if it goes through again, each of those will produce 2 identical daughter cells, and so on and so on and so on. Okay, just a little thing, this G0 phase, some cells actually undergo what we call a gap 0 phase, where it just keeps growing and doing its normal cell functions for the rest of its life. It doesn't actually cycle through the rest of the cell cycle. Okay? All right, let's actually go through each of these processes. The first one we're going to talk about is what we call interphase. An interphase is actually the G1, the S, and the G2 stages of the cell cycle. All right, and even though mitosis is extremely important, the eukaryotic cells actually aren't usually dividing. Usually they're in this what we call interphase that's made up of these 3 components. G1, cells go through day-to-day routine, growth, and all that stuff. G2, also day-to-day routine, just we're going to have double the chromosomes, because during synthesis, this is what happens here, all right? The chromosomes go from single-stranded chromosomes to double-stranded chromosomes. It's important to note that each one of these strands of chromosomes is identical to that strand there, okay? This is what a cell looks like during interphase. Nothing too exciting. We can say a nucleus with just some squiggly material there. We can't see the chromosomes in themselves. After interphase, once the cell decides to go through mitosis, it has first to go through this first stage of mitosis, which we call prophase. Pro coming from the word before. Okay. So during prophase, the chromosomes condense, and they become more visible. I think of condensed milk, which is nice and thick and really goopy. All right, the chromosomes do that. They become nice and thick. Okay, the spindle fibers, which are going to attach to the chromosomes, are going to form from these structures that we call the centrioles. Okay. And also the nuclear envelope, so the actual wall of the nucleus, starts to degrade so the cell can actually do stuff inside of there. This is a really clear cell going through prophase. We can see that because as much as we still have that nuclear envelope around here, the chromosomes are really clearly visible. They're condensed, okay? After prophase, we have what's called metaphase, and in metaphase, those spindle fibers, which are these cytoskeleton proteins of the cell, they attach to the chromosomes and they line the chromosome up along the midline of the cell like this. Okay, same thing's happening in here in this real cell. We have the centriole structure there, all right, and you can see these fibers that are attaching to the chromosomes, and these fibers hold the chromosomes along the midline of a cell. This is one whole cell over here. Cool. And then we're going to actually pull these chromosomes apart, okay? So what happens is the spindle fibers will pull the sister chromatids apart, and I'll tell you what the sister chromatids are in a second, they pull them apart and move them to opposite poles of the cell. So a set of the sister chromatids goes to one side of the cell, and the other set goes to the other side of the cell. What the sister chromatids are, if we go back here, is each one of these identical strands of the chromosomes is called a sister chromatid. After anaphase is the final stage of mitosis, which is called telophase, and all really telophase is the nuclear membrane of a cell now reforms. We're going to have a nice nuclear membrane, all right? Those chromosomes are probably also going to start to decondense, all right? And we might actually see a bit of pinching of the plasma membrane, especially if you're talking about an animal cell like this. If it's a plant cell like this image here, you're actually not going to see that. But the cell membrane is still fused together. It's still technically one big cell, okay? Because after mitosis, we have what's called cytokinesis. And what cytokinesis means is cyto comes from the Latin word for cell, and kinesis means to break. So pretty much cytokinesis is the process that breaks a cell. Okay, the cell membrane will actually pinch, all right, and through that pinch, the cytoplasm of each of the cells will separate, and you're going to get two brand new cells, just like you can see here. It's really cool. Radioactively marked cells. You can watch the process of cytokinesis and splitting between two cells. So it's really important that you can, through the process of mitosis, understand to be able to identify mitosis in both an electron microscope image like this and also in handmade images. So I've identified 4 different cells here. See if you can identify which is which in terms of prophase, metaphase, anaphase, telophase, and cytokinesis, what I like to call PMAT plus C. Alright, so have a go at doing the same for this one here, identifying these 4 cells in terms of what part of the process they are undergoing. And we'll have a look at those together in class.